What is fermentation? This is a bioreactor. Do you know what's happening inside? Microorganisms are growing and dividing, and as they do so, they're producing the active ingredient of a biopharmaceutical. This module describes the significance of microorganisms in biotechnology, explaining how they can be used to produce therapeutic proteins. It also explains the technologies used in a fermentation process and how the process can be optimized. Other areas that you will cover include the cultivation of microbes in bioreactors, the nutrient media used to grow microbes, microbial growth kinetics, and sterility. Fermentation is one of the most important processes used in the biopharmaceutical industry. Additional factors for successful fermentation. Apart from sterile media and equipment, there are additional factors that must be considered to ensure optimal fermentation. Water, gases such as air or oxygen, acids and bases for pH adjustment, and foam depletion agents may need to be added. Oxygen is supplied by filtering air, and sometimes carbon dioxide is supplied, depending on the nature of the fermentation. pH is usually controlled by the addition of phosphoric acid, which decreases the pH, or the addition of sodium hydroxide, which increases the pH. The production of foam during microbial fermentation is a serious issue that must be addressed in the majority of fermentations. Foaming occurs due to the agitation of the medium and the addition of various components to it. The foam produced can damage the cells and cause fouling of the equipment. Foaming is prevented by adding chemical anti-foam agents and by using mechanical techniques. Sometimes the gene coding for the protein of interest, in other words, the product, will only be expressed in response to the presence of a certain compound known as an inducer. The inducer is included in the growth medium. An example is starch, which triggers the production of amylase, which is the enzyme required to break down starch.